let's go ahead and get started. So ESMFold. ESMFold is part of the ESM family, which is a family of language models, mostly. So ESM was originally started by a group at Facebook AI Research. Eventually, they split off, and now they're on their own company called Evolutionary Scale. And Evolutionary Scale just really released ESM3 recently. But there's a bunch of different models of different sizes as well. So we have ESM2, which is a language model. We'll talk a little bit more about that. It's a protein language model that can predict function, properties, as well as other unsupervised tasks. ESMFold, which we'll be talking a little bit more about now, a little bit later actually. ESMMSA, which can extract embeddings from a multiple sequence alignment, which is a little interesting. ESM1V, which was trained for variant prediction effects. And then ESMIF1, which is an inverse folding model, kind of like protein and PNN that we'll talk about a little later. The latest model, ESM3, is a little bit more unique than its relatives because it simultaneously reasons over structure, sequence, and function, which is really unique. So we're not going to talk about that, but it's a pretty cool model if you want to check it out. So let's talk about ESMFold. Like AlphaFold, the main goal is given a protein sequence, what is the structure? But it's a little different in the sense that we don't use an MSA, we don't use templates, we use the protein language model, or PLM. Once again, you get the output structure with the confidence values, as well as a pairwise confidence as well. So this is like kind of the main difference between these two models. AlphaFold follows this upward track where we have this multiple sequence alignment that really gives evolutionary history of this protein, maybe some information about coevolution that can give some insight into how this protein is actually structured. Then you have, your structure module and you get your structure. The difference with ESMFold is we throw it through a transformer-based language model that gets embeddings for that sequence. And then we just use those high-dimensional embeddings that represent the sequence and ideally some sort of evolutionary information, although like what actually is there is kind of hard to tell sometimes. But we take these embeddings, throw that through a structure prediction, or through some structure modules and get a structure out. So quite similar, but the main difference is that language model. So why use a language model? Well, we have so much more sequence data than structure data. It's just way more abundant. We have billions of sequence data compared to the 200,000 structures that we have in the PDB. So it's just way more plentiful. And language models are super well developed and pretty mature at this point, as you've seen probably use ChatGPT, etc. But the whole premise is kind of you give it a sequence that is partially masked, as we have these little white squares. And then you ask the model what should be there, what fill in those blanks. And so after training a while, it should be getting good at filling in those blanks. It's kind of like recognizing sequence patterns in the protein universe. And then it has some sort of idea about evolutionary history because it's seeing similar proteins as well. So it should be able to fill in those gaps and start learning what a protein sequence looks like. And once you've done this, you can kind of take these embeddings and use them in a variety of other tasks, which are super cool and super useful. Essentially, these become features of your sequence. And you can use them for downstream tasks like contact map prediction, secondary structure prediction, mutational effects, function, as well as sequence design. As we've seen in those other ESM models, they've all kind of started with a language model. So first of all, let's talk about the performance. It's pretty good. It compares slightly worse than AlphaFold and RosettaFold when they have an MSA. However, when they don't have an MSA, ESMFold is much better, primarily because it doesn't need an MSA, so it has all the evolutionary information it needs in the protein language model. Whereas, as we saw with AlphaFold, it's heavily dependent on that size of the MSA. As you can see, AlphaFold full is better, and ESMFold is better with no sequence. The confidences correlate, which is reasonable to see. Yeah, that's just reassuring. Let's talk about the actual model. So really, they take your single sequence, your input, your query sequence, throw that through the language model. This is already pre-trained. We don't really fine-tune this at all. But they have ESM2. They throw it through, get those embeddings for that entire sequence, throw it through a folding trunk, which is kind of similar to the EvoFormer, as we'll see. Throw it through a structure module, which is identical to the structure module in ESMFold. And then we get the predicted structure out as well as the confidences. They also do some recycling as well. So this is what the folding block looks like. Quite similarly, we take in a sequence representation rather than an MSA representation. And then we have our pair representation. They both update each other, so we have some self-attention on the sequence. This replaces that column and row attention in the EvoFormer. We don't really need that anymore because we don't have an MSA. And then the pair representation also has the triangular updates that AlphaFold uses. So it's really quite similar to the EvoFormer. Just if you want that comparison, here it is. Maybe, yeah, so self-attention replaces the row-wise and column-wise attention. Transitions quite similar, outer product mean. And then the triangular updates for the pair representation. And then you have your updated representations.
Maybe I missed it, but where does the pair representation comes from? Before it was from templates, right?
Yeah, that's a good point. So they simply just like do a outer product to get that L by L by C.
But just the sequence.
Well,
or
I think is it based on the sequence or is it based on the embeddings? I think it's based on just the sequence alone, but the embeddings might. I can't remember exactly. I'm sorry. Yeah. Okay. Like I said, structure modules completely identical. Yeah. So that's identical. The loss is very similar. We have pretty much the same components except for the masked MSA loss. So you can imagine they're kind of teaching the model pretty similar things. Yeah. Don't really think it needed to go into more detail. Similar limitations to AlphaFold. Although the de novo proteins and the mutations are less of a problem with ESMFold, although I don't think that problem's solved. So while it's better, I still wouldn't necessarily trust those predictions. Similar results, multiple states can't really trust, and it doesn't really explicitly model the other. Similar to AlphaFold, they've also done ESMAtlas, which has predicted a ton of structures, over 700 million, and a lot of them are high confidence, which is really cool to see. These are from a lot of metagenomic sequences, so necessarily from like the environment that kind of just, yeah. So that's a nice resource. They've also extended it to a programmable design language, which I'm not sure many people use, but I think it's a cool extension of kind of combining the language model constraints and the ESMFold constraints into a design algorithm. Yeah, I thought it was cool and thought I would share it. And then the last thing I really wanted to talk about was ESM3, which is maybe not the direct extension of ESMFold, but it's very similar in the sense that it also can predict structure. However, it's predicting structured tokens, which is slightly different. And we could talk more about that if you're interested, but it follows a very similar pattern where you're kind of trying to predict the mass tokens, and then you can also get structure out, as well as function and sequence, which is kind of cool too. So that's the most recent one, just came out, I think in July or something like that. But yeah, I think that's pretty much all I have for ESMFold, because it's quite similar.
Sorry, what guy you were?
Yeah, so it predicts like residue level annotations for the function. So whether this is like a glycosylated residue, whether it is post-translationally modified, it's an active site, something like that.
So what's your alcohol rate? Even though it's not modeling the molecule or other things that it needs space for.
Yeah, it'll do pretty much the same thing.
So it will say it's an active site, but not co-op, right?
Yeah.
Okay.
Yeah. But you can also condition it on the input function. So if you want to say, I don't know, I want to make a hydrolase, you can tell it that, and then it should make something that is hydrolase-esque, which is, it's kind of cool. They used it to design a new GFP, which was quite different from native GFPs. So.